Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about the study of microbiology and how it has transformed our understanding of life and has led to major advancements in the biomedical industry. Thank you so much for being here. Let's get started. So there are different subdisciplines of microbiology. Microbiology is the study of small life. Micro means small. Biology is the study of life. And so when we're talking about microbiology, we are studying tiny life. But there are different subdisciplines. There's bacteriology, that is strictly the study of bacteria. If you take a microbiology course, that is where the general focus lies. But there are other interesting subdisciplines like mycology, the study of the fungi, and then virology and parasitology. I've taught both of those classes in the past as well. Virology is the study of viruses, and it's really kind of like an intense applied molecular biology, and it's awesome. Parasitology is the study of parasites, often single-celled organisms, but we go into multicellular organisms as well. Things like tapeworms and giardia, really interesting infectious agents. So throughout most of human history, a lot of people believed in a concept called spontaneous generation. This was the idea that life could come from non-living things. And a really good example of that is right here on your screen. People thought that meat, food, could give rise to life. And one of the examples was that meat would eventually give rise to maggots and flies. And there were some scientists who questioned that, one of which was Francisco Redi in the 1660s. He decided to do what seems today to be a rather simple experiment, but it was hugely important. He questioned whether or not spontaneous generation could actually happen. And what he hypothesized was not that the meat, the rotting meat at this point, was giving rise to maggots and flies. It was that, well, people were leaving out the meat and flies were coming in and laying their eggs in the meat, and then the meat would eventually allow a great food source for the eggs to hatch and maggots to feed upon. And so what he did is he ran a simple experiment and he covered the meat, and he showed meat that was left uncovered would give rise to maggots because the flies were indeed laying the eggs in the meat, and the meat that was covered, well, the eggs didn't have contact with the meat, and therefore the meat did not give rise spontaneously to maggots. And so that was, generally speaking, one of the first attempts to disprove this idea of spontaneous generation. But these ideas sometimes can be strongly held, and it can take a while for them to, well, be proven wrong in at least most people's minds. And so it actually took a couple hundred more years for another scientist to come along and do another really important experiment. Now, around that same time, Antony van Leeuwenhoek was doing his work, and one of the things that he was working on was a way to see microbial life. And what he did is he developed one of the first really good microscopes. Now, this is going to look a little bit different than what you and I are used to, but it was really important because now you could visualize these things that scientists said, hey, these exist. Nobody could see them. It's almost like a ghost story, but he was able to visualize them. And this is a representation of one of the first microscopes that Leeuwenhoek developed. And basically what you had is you had that little needle on the end where you could put your specimen, and there was a very small magnifying glass at the end of it. Now, this was different than anything else that had been developed in the past because the magnification was much better, and he was actually able to see microbial life, things like protozoans. So we fast forward a couple hundred years, and we have this wonderful human being, Louis Pasteur. Many of you know his name because of pasteurization, using heat to control microbial growth in food products. But there was something else that he did that was really important in the history of microbiology, because even in the 1800s, 200 years after Redi did his work, spontaneous generation was still a widely held belief. And part of the reason for that is scientists were now learning how to grow microbes. And when you grow microbes, you need special solutions. And sometimes scientists would make these special solutions, not add any microbes, and spontaneously microbes would appear. And so they were saying, hey, this is an example of spontaneous generation. But Louis Pasteur, he had different ideas, and he wanted to show that, no, spontaneous generation isn't a thing, even in microbial solutions like a microbial broth. And so here's the experiment that he did. So his hypothesis was simple. If you had a solution and you heated it up and you sterilized it via that method, cells, as you can see at the top on the right-hand side, would enter through the mouth of that flask. They would enter and they would propagate and they would grow. And so what he was trying to show was, listen, It's not spontaneous generation that's happening here. It's actually just airborne contamination, something that seemed a little bit odd back in the 1800s, and it's something that we take for granted today. And so he was able to show, hey, I can sterilize this. I believe that it's cells coming in through the top. Now, how did he prove this? Well, he developed something called a swan neck flask. The swan neck flask was designed to keep bacteria out of the solution, out of the broth. And so what he did is he boiled the broth once again. The cells would get caught in the swan neck flask and not actually enter the solution. And he was able to show, hey, what was happening was not spontaneous generation. It was actually airborne contamination. And for the most part, that was the final nail in the coffin of spontaneous generation. And he was able to show, you know what, if I go ahead and I tilt this flask and I expose the broth to the cells that were caught in the swan neck, growth is going to happen again. Now, even with that, sometimes the broth would still get contaminated even if he didn't tip the flask over like you're seeing here. And one of the reasons for that was, well, it turns out the way the broth was actually being manufactured. So around that same time, another person named John Tyndall was working in the same area in microbiology, and he was able to show, along with Ferdinand Cohen, that there's these other infectious entities that are basically metabolically inert until they enter a broth solution. And they can be airborne, and they can actually survive the heating process, and these things are called endospores. In an endospore, basically what happens is some microbes are able to create this protective coating around their cell and become metabolically inactive. And these endospores are really, really resistant to heat and UV light and desiccation, which is a fancy way of saying drying out, and they can remain viable for dozens of years, maybe even more than 100 years. Now, the problem was when they were creating this broth, they were actually, in some cases, using things like hay extract that had endospores contained within that extract. And then once the spores were in that solution, they would come and become a vegetative cell and they would grow. And so it looked like there was contamination even with the swan neck flask, but really it was the spores in the broth from the beginning. So I mentioned this golden age of microbiology. It was a very exciting time to be somebody who studied microbiology. It was basically from the mid or so 1850s through the 1910s, maybe close to 1920. And during that time, the reason why it was called the golden age of microbiology is there was so many discoveries that were happening. They had discovered many of the pathogens, microbial pathogens, that were causing disease at the time. In 1900, they discovered this concept called a virus, which was even smaller than bacteria. You couldn't even see it. And during this time, they were beginning to learn how to avoid these infectious agents and how to remain healthy in their presence. And I think this is a really important figure. This is from the Center for Disease Control, the CDC. And what it's showing is the leading causes of death in 1900. And in this particular picture here, they're showing 1997. So through a 100-year span throughout the 1900s. And I'm going to show you an updated version here in a second. But what we can notice, hopefully what we see are the trends. Look at 1900. Look at the top 10 leading causes of death. You have things like pneumonia and tuberculosis and all kinds of infectious disease. And you can see those were the primary leading causes of death in 1900. And by the way, the average life expectancy in 1900 was in the 40s, low 40s. The reason for that is there were a lot of infant mortality going on, a lot of infant deaths. And so you fast forward almost 100 years, and what we see is a lot of those infectious agents, those diseases are gone. And so we have things like now heart disease and cancer and stroke, chronic lung disease, diabetes. And you see far fewer infectious diseases as a leading cause of death almost 100 years later. Well, let's go ahead and fast forward. So now we have 2017 and 2018, and we can see the leading causes of death once again: heart disease and cancer, unintentional injuries, lower respiratory diseases, stroke, Alzheimer's disease, diabetes. And so you can see here the only infectious agent on here is influenza, and it's down 1, 2, 3, 4, 5, 6, 7, 8, number 9. What has happened over the last 100 years that has greatly reduced the number of deaths due to infectious disease? Well, again, this is from the CDC, and they show some of the major developments that have happened over the last 100 to 120 years. And you can see things like, well, for one, keeping our drinking water away from our wastewater. That has been huge. So when we're looking at the major discoveries, I would say that's definitely one of them. Drinking water and wastewater are separate. The second thing, and you can see here the advent of things like penicillin and antibiotics. Because when people contracted bacterial infections before then, there was really not a whole lot you could do other than supportive care. But for the first time in the 1940s, we now have something that can actually destroy microbes in the human body. Vaccination also was a massive development. And so now not only could you treat somebody who had an infectious agent, but through vaccination, you could prevent them from ever contracting the infectious agent in the first place. Other things, certainly just general hygiene, washing hands, keeping surfaces clean. In a hospital setting, the nosocomial infections, the hospital-acquired infections went down dramatically. Before the advent of aseptic techniques, you were just as likely to die from surgery as the original injury that causes surgery or the original illness that causes surgery in the first place. And so once we developed things like sterilization and aseptic technique, the number of infectious agents and the infections in a hospital setting went down dramatically. This is a wonderful map of emerging and re-emerging diseases. These are diseases that had not previously infected humans, but have gained the ability to do so. And we can see here, there are all kinds of diseases, things like Ebola viruses and SARS viruses. You've got bacterial infections as well, things like MRSA and C. diff. There are so many different types of diseases that can actually come from other animals. Usually an emerging disease comes from a different animal, gains some ability, some genetic mutation that allows it to infect humans, and that is usually how it winds up in a human population. One of the most important is influenza, and you can see all the influenzas over here on the right-hand side. Many of them are actually viruses, influenza viruses that infect things like birds or pigs, and then through genetic mutation, things we'll talk about later on in a future video, they're able to infect human beings and then spread. Now, emerging diseases can be dangerous because they can cause epidemics, localized widespread infection, or pandemics, which can be worldwide. One of the things we talk about a lot in microbiology is, yes, we are used to hearing about microbes that cause disease and make us sick, but microbes are absolutely critical in our everyday life as well, both in our bodies and environmentally. And in our bodies, we have microbes. We call them normal flora or normal microbiota that live on the surfaces of our body all the time. And what they do is they do things like prevent infection by taking up binding sites. And later on, we're going to learn that one of the first things that an infectious agent has to do in order to cause an infection is attach. And if you have these microbes that don't harm us, attached to our surfaces of our body, the pathogens have nowhere to call home. They can't attach. The next thing that they do is some of our normal flora, the microbes that are always associated with our body, can actually release antimicrobial chemicals that destroy pathogens. They also help produce certain vitamins for us, and there have been studies that have shown that some of the metabolic byproducts and some of the things that they produce actually improve our cognition. They will also alleviate some autoimmune diseases, and they help potentially control the body mass index, BMI. There are environmental benefits as well. Microbes fix more CO2 than all the land plants combined. They also produce more oxygen. Microbes, in particular cyanobacteria, fix nitrogen. This is hugely important. We're going to talk about the nitrogen cycle later on in a future video. But without the microbes fixing nitrogen, things like amino acids and proteins, well, not sure they would really exist. Microbes also associate themselves with plants and plant roots, and they have a symbiotic relationship where the microbes provide things like nitrogen-based compounds to the plants. The plants provide the microbes sugar, and it's a great relationship that helps the plants grow stronger and faster. These microbes may help alleviate some plant diseases or prevent them. And they're really important. The bacteria are really important for things like bioremediation. We've learned how to take microbes and break down environmental contaminants, things like oil and some pesticides, even nuclear waste. So that is a very brief history of microbiology, what microbiology is, the golden age of microbiology, how microbes can make us sick, how they also benefit us. As always, thank you so much for being here. Have a great week, everyone.